Hello everybody, my name is Esmaeil from Janssen Lab, University of Copenhagen, and today I'll be talking about extracting disease-lifestyle relations from biomedical literature. As we all know, disease onset and development is not only related to genetic factors, but also closely related to lifestyle factors, such as physical activity, nutrition, smoking, among others. About genetic factors, we are in a pretty good situation, and there are a lot of databases which covers gene disease associations. But this is not the case for lifestyle factors, and there's no knowledge base for them. So disease lifestyle associations are still hidden in scientific literature. There are more than 35 million abstracts in PubMed, and this is just the visible part of iceberg if we consider full text articles. No matter if you consider only abstracts or full text articles, they are too much to read. So we need text mining. The aim of this project in the first place is to detect lifestyle factors and diseases from literature, and then finding the associations between them, and finally creating a knowledge base like this, which covers different types of associations and relations between diseases and lifestyle factors. And as you can see, the only type of relation between disease and lifestyle is not the causal relations, and we can have different types like treatment or prevention or other types of relations between these entities. There are three different steps to be able to create such a knowledge base, starting from named entity recognition, followed by relation extraction, and then using all those extracted relations to create a knowledge base. Let's go to the detail of these steps. First, starting from named entity recognition, which is basically detecting lifestyle factors and diseases in the text, like this example. About detecting diseases, thanks to disease ontology and similar resources, there's no problem, and we can just easily tag and detect these mentions in the text. But this is not the case for lifestyle factors, and there's no available ontology or dictionary for lifestyle factors, and even the definition of lifestyle is not clear. That's why we started creating a lifestyle factor ontology for the first time, and of course the prototype version just using brainstorming and some existing ontologies for nutrition, exposure, and smoking, and some of available questionnaires. This resulted in the first version of the ontology, which covers 9 different aspects of lifestyle and around 900 concepts. Then we decided to expand our ontology and add new lifestyle-related concepts to the ontology. But how? We did this in 2 different steps and using 2 different approaches. In the first step, we used Wikipedia titles to see if they can be lifestyle-related concepts or not. The reason for this assumption was the fact that most of the existing concepts in the prototype ontology were among the Wikipedia titles. So why not to check the other titles? We trained the BioBERT model to classify all the Wikipedia titles and add those which are classified as lifestyle related. In the second step of ontology expansion, we considered many existing knowledge bases and not only Wikipedia titles, and also more than 1,000 existing ontologies are queried for lifestyle related concepts. And this resulted in tens of thousands of candidates that can be a lifestyle factor. Clearly, we need filtering to see if these candidates can be added to the ontology or not. we use two different metrics to apply filtering on the candidates, which are somehow exploitation and exploration. Because we are interested to find synonyms of existing concepts in the ontology, which is the exploitation part, but also we are interested to find new concepts which are missing in the existing ontology, and that's the exploration part. So we gathered existing concepts from the ontology, as you can see here, plus candidates that we are queried from different resources and knowledge bases and existing ontologies, and we want to filter them. And then we converted all of them to embeddings, as you can see, and it's visualized here in this plot. We use two different approaches to fulfill our two different metrics. The first approach just calculate the distance of the candidate to the nearest neighbors in the ontology and assigns a score based on this distance. But the second approach uses a topic modeling method to see if this candidate belongs to lifestyle related topic or not, and then assign a score based on that. And then we combine these two scores and use the calibrated combined score to filter the candidates. Even after automatic filtering, we need manual checking before adding these candidates to the ontology. That's why we created this interface to help the curator to see if the candidate should be added to the ontology and where it should be added and what is the correct place. So these candidates are provided along with all the information from sample sentences in PubMed or similar or nearest neighbors in the existing ontology to help the curator to do this. So this step is the final manual check before adding them to the ontology. And this is the final version of the ontology after initial prototype creation and then two different steps of semi-automatic expansion. These are statistics of 9 different branches of lifestyle factor ontology and also statistics of the all names including synonyms. And this is the word cloud of some of the existing concepts in the ontology, color-coded based on the branch, and also the size corresponds to the frequency of the name in the literature. Okay, now that we have the lifestyle factor ontology, we can use it to finalize the first step, which was named entity recognition, and we used Tagger, which is a dictionary-based NER tool developed at Jensen Lab to detect both lifestyle mentions and disease mentions in the text. As you can see, Tagger receives biomedical literature and both lifestyle and disease ontologies and is able to tag or detect both lifestyle mentions and disease mentions in the text. Using our comprehensive lifestyle factor ontology, we were able to match millions of lifestyle related mentions in the text, which is really awesome. And you can see the statistics categorized for different branches. Now we are ready to do relation extraction. And the first and the straightforward approach to do the relation extraction is called co-occurrence-based relation extraction. Co-occurrence-based approach simply counts if a pair of disease and lifestyle are co-mentioned in the same context, let's say same sentence or paragraph or document, and counts these co-mentions. Using this simple approach, we were able to find more than 500,000 associations between disease and lifestyle factors. Using co-occurrence-based approach, we can create nice plots like this, which shows how different branches of lifestyle factors are associated with diseases in general here, or different branches are associated with a specific disease. For example, in the case of nutrition, you can see significant contribution of nutrition for obesity and diabetes, which makes sense. But the co-occurrence-based approach has drawbacks, and you cannot distinguish the relation type between disease and lifestyle. But we are interested to classify different relation types between disease and lifestyle factors, other than causal relationship. For example, lifestyle can prevent from a disease or even treat a disease. As you can see here in this example, even between same pair of entities, there can be different relation types, but this cannot be detected by the co-occurrence-based approach, since this approach simply ignores the context. That's why we need better approach. We fine-tune the BioBERT model to be able to distinguish between different relation types. As it is expected these days, we started from a pre-trained language model, which is BioBERT in this case, and then we used a custom relation extraction pipeline, and we fine-tuned this model to be able to distinguish between different relation types. To fine-tune the model using supervised approach, we need labeled samples, and of course we don't have labeled samples off the shelf in this case, so we started manual annotation to provide labeled samples for fine-tuning the model. For manual annotation, we started by developing the annotation guidelines, and of course we did several rounds of inter-annotator agreement to be sure about the developed guidelines, and then we moved forward for the rest of the annotation. We annotated 400 abstracts using the developed guideline, and then we fine-tuned our model using these samples. As you can see, performance evaluation results for distinguishing between 4 different relation types between disease and lifestyle factors is really promising. And even we want to go further and distinguish between positive and negative statistical associations, because it is important, and the positive association means increasing the risk of a disease, and negative association means decreasing risk of a disease. And also we want to distinguish between prevent and treat relation, and then finally we will have 6 different relation types. But to be able to classify these different types and increase the number of classes, we need more samples, more labeled samples. And this is the next step of the project, to expand the annotated corpus, to train more generalized model to be able to detect fine-grained different relation types from the literature, and then create a knowledge base from all these relations. Of course, we want to use this process not only for biomedical literature, but also for patient records and electronic health records, which is really useful. It was all from me. I want to thank all the collaborators in this project, and thank you for your attention.